Biology in Tamil Korvai channel சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். In this video I am going to teach you about reproduction in organisms. This part 2 video, part 1 video already published. The question paper also sent to you. You would have attended it. And this video also going to bring a question paper, both multiple choice question and also descriptive, and please attend it and get the full mark. I wish you all the best for getting full mark. To get full mark, I request you to stay with the video till the end so that you are able to understand all the concepts thoroughly. I hope you will like this video, students. If you like this video, press the like button, share this video to your friends, comment on it, subscribe our Biology in Tamil Korvai channel, press the bell icon to receive the notification. Now I want you to open the book, textbook first. Please open the textbook and open one notes taking notebook and also a pen, so that as and when you wish to take notes, you can take the notes for your future reference. Now let us enter into this video. And this is the overall flowchart which I already sent. In our book also the flowchart is given. This flowchart also very helpful for you. And here already we covered in video 1 reproduction, definition, asexual reproduction, sexual reproduction and fission. All the types of fission we covered in the video 1. In this video we are going to cover budding, sporulation, regeneration, fragmentation, gemmule formation. So these are the 5 subtitles we are going to cover in this video, students. Now first one is budding. Budding in sponges. It can be asked in 3-mark question. So you can take note as it that is a 3-mark question. Budding in sponges. Budding. In budding, the parent body produces one or more buds. You see, these are called as buds. Like flower buds, the same way here also buds are produced. Each bud grows into a new young individual. It means daughter individual. So it is in sponges. Here it is given budding in Leucosolinia. The sponge name is Leucosolinia. The sponge belongs to, very well, the Porifera, the phylum Porifera. Likewise, you can at least relate it with one additional concept which you already know about the sponge, student, so that this concept you will not forget at all. The buds separate from parent to lead a normal life. The buds separate from a parent to lead a normal life. In sponges, the bud constrict and detach from the parent body, and the bud develops into a new sponge. The bud develops into a new sponge. So this is the place where the constriction occurs. This is the place where constriction occurs. The word constriction means the place of detachment. The detachment of the daughter individual from the parent individual. Budding in Hydra. So budding in sponges we learnt. Now it is budding in Hydra. This can be asked in 5-mark question, students, including the diagram. When buds are formed on the outer surface of the parent body, the buds are formed on outer surface of the parent body, it is known as exogenous budding. Example, Hydra. Exogenous budding. What is exogenous budding? 1-mark question. If it is formed from the surface, it is called as body surface. It is called as exogenous budding. Budding in Hydra continues. In Hydra, when food is plenty, here also you have to stop and remind yourself that you should remember this concept for 1-mark question, because one question can be asked. The budding in Hydra take place during unfavorable condition, when the temperature is high, when the salt concentration is high. Likewise, they can create the option. Along with this option, along with those option, this is also one of the option. Food is plenty. So please take note of it. Budding take place during a very favorable condition when the food is plenty. It means it can produce new individuals in order to eat the additional food available in the surrounding. That is the meaning. The ectoderm cell increases. The ectoderm cell increases. This is the ectoderm. Red line is ectoderm and blue line is endoderm. And the cavity here is called as gastrovascular cavity. Okay. Now the body surface. Ectoderm increases and form a small elevation on the body surface. Ectoderm and endoderm are pushed out to form the bud. The ectoderm and endoderm pushed out to form the bud. You see the bud. The budding in Hydra, the bud contains an interior lumen. The bud contains interior lumen. This is the interior lumen which also extend within the bud. Now this is the gastrovascular cavity. So lumen means the space enclosed within a certain structure. Then it is called as lumen. In continuation with the parent's gastrovascular cavity. So in continuation with parent's gastrovascular cavity, right? The bud enlarges. The bud enlarges, develops a mouth. You see the mouth and also the tentacle in its free end. When fully grown, the bud constrict. Again, the word constrict comes. Constriction take place here at the base and finally separate from the parent body and leads an individual or independent life. So this is about budding in Hydra. So intensive learning is required. Both intensive and extensive learning is required, student. Intensive learning means taking one concept. So they are giving ectoderm and endoderm. Now you have to understand what is ectoderm, what is endoderm, what is the lumen and what is the gastrovascular cavity. So all these informations are given in our 11th standard book also. Okay. So you have to understand and proceed with the learning. Then it is called as intensive. Okay. Extensive wide learning. So both are required for preparation towards the competitive exam, students. Now one more intensive way of learning. Okay, exogenous budding, then naturally opposite endogenous budding. So the difference can be asked in 2 mark or 3 mark, students. What is exogenous budding and what is endogenous budding? Exogenous budding, as already we discussed, surface, it arises from the surface of an organism, example Hydra as well as yeast. Whereas endogenous budding, okay, it is a bud, gemmule formation that we are going to learn, student, that is which occurs in the sponges. So sponges already we learned, it is for budding. Now in a future slide we are going to learn about the gemmule also. So that is considered to be as endogenous budding, student. Develop inside the body of an organism. It develop inside the body of the organism. Example sponges given here, and one more example also we are going to learn. That is Noctiluca. Okay, Noctiluca. This is for endogenous budding. In Noctiluca, hundreds of buds are formed. You see, this is the Noctiluca structure, and within the cytoplasm, you can see many buds are produced. Okay, hundreds of buds are produced inside the cytoplasm, and many remain within the body of the parent. This is called as endogenous budding. So this is one of the structure which I drew for you. That is the Noctiluca. So this can be asked in write note on Noctiluca in 2 mark question. Now gemmules, gemmule formation. So this is one of the exclusive type of reproduction to overcome the unfavorable condition, student. Already we learned Hydra is producing the bud during the favorable condition, during the availability of more amount of nutrients. Now it is during the unfavorable condition. The freshwater sponges and in some marine sponges also. You see the word freshwater sponges and in some marine sponges. So this can be one MCQ. Okay, by changing the option. Regular and peculiar, again 2 conditions given. Regularly, in addition to the regular reproduction method, peculiar mode of asexual reproduction occurs in freshwater sponges and marine sponges. By internal buds, here also they are giving, you see, internal buds. So you can consider it as an endogenous type of budding called as gemmules. So this is the gemmule structure. This is the gemmule structure in sponges. You see gemmule in sponges. So this alone can be asked in one of the diagram question. Draw and label the gemmules, or they may give the diagram and asking you to label the parts. So inner membrane, then archaeocytes, these are the very important cell to produce the new sponge. Outer membrane, this is the outer membrane. Micropyle, you see this opening is called as micropyle. You see the micropyle, I would like to connect the concept with the ovule. In ovule also the opening at the one place of discontinuous region of the integument is called as micropyle. So that is also opening, this is also small opening, micropyle. Now you can connect these two words so that you are able to remember. So these also cells, okay, these are called as archaeocytes to produce the new sponges. And here, in order to protect these that is gemmules, the spicules are present. Spicules are like a spine-like structure. These are called as monoxon spicules. These spicules are called as monoxon spicules. So this is one type of spicule, student. So there are other type of spicules also. That is, I think, beyond our scope of learning this reproduction. Now, a completely grown gemmule. A completely grown gemmule is hard ball. Is a very hard ball because it is going to face the unfavorable condition. You know, so that is the reason why it is a hard ball, consisting of an internal mass of food-laden archaeocytes. These are called as archaeocytes. Food-laden archaeocyte, because after the return of favorable condition, it has to produce the new sponge. That is the reason why it is laden with food. During unfavorable condition, the sponge disintegrates, but gemmule can withstand the adverse condition. When condition become favorable, the gemmule begins to hatch into new sponge. Now, the fragmentation can be asked in 3-mark question. In fragmentation, the parent body breaks. As the name implies, the parent body breaks into many pieces, and each piece develops into a new individual. So, example they gave for pedal laceration in sea anemone. Pedal laceration in sea anemone. So, this is the pedal laceration, fragmentation or pedal laceration. So, the question can be asked, pedal laceration occurs in many genera of sea anemones. Many genera of sea anemone. It belongs to the phylum called as Coelenterata. So it shares the phylum along with the Hydra, students. So that also one of the connectivity you can establish. So now you can see this is the pedal disc at the base. It consists of pedal disc. You see the Hydra consists of basal disc. Here it consists of pedal disc. Again one more connectivity you can establish. So that whenever the question is asked between pedal disc and basal disc, you will not forget which one is applied for which organism. So pedal disc for sea anemone. Now this is the pedal disc by which it is able to attach itself with the substratum. Are you clear? So it produces small bud-like structure. You see, this is called as pedal laceration, and the constriction occurs due to the constriction. Okay, this there is a small bud-like structure is detached from the parent body, and that grows and develop the tentacles and produce a new individual. So this is called as pedal laceration. Lobes are constricted. These lobes are constricted again. So third place the word constricted is given. Constricted off from the pedal disc, and each of the lobe grows mesenteries. So mesenteries are nothing but this, that is the partitions within the body. Within the body is called as mesenteries. The partition and tentacles also arise to form a new sea anemone. So having mesentery is one of the characteristic feature of Coelenterata when compared to the sponges. Okay, so this is the pedal laceration occurs in many genera of sea anemone. The enlarged view. Okay, next fragmentation continues in the tapeworm. Tinea solium, scientific name of the tapeworm. The gravid right proglottids. So these are the gravid right proglottids, or the oldest at the posterior. Oldest at the posterior, and they are new at the anterior region. Okay, posterior end of the strobila. Posterior end of the strobila. You see, this is the structure. Okay, Apolysis in Tinea solium. This is the diagram given in our book. Scolex, the head-like region, followed by the neck. immature proglottids, and at the posterior region mature proglottid, and still at the end it is called as gravid proglottid, which carries the reproductive structure. Okay, so it detached from the parent body and go along with the fecal matter. The gravid proglottids are regularly cut off either singly or in groups from the posterior end by a process called apolysis. So what is apolysis? The question can be asked. So this is the word which is exclusively applied, okay, in our textbook with respect to the tapeworm. Please remember it. Okay, due to the apolysis, the proglottids, mature proglottids or gravid proglottids are coming out and it is eaten by the pig, so that the connection is established between the man and pig. So it is very significant in the life cycle of tapeworm. This is very significant since it helps in transferring, you see, transferring the developed embryo from the primary host man to find a secondary host pig. Okay, so what is the significance of apolysis? They can ask. So it is transferring, it is completing the life cycle of the tapeworm. That is the answer that you can give. Regeneration. Now we are going to learn about regeneration. This can also be asked in 5-mark question because it consists of, there are two types, morpholaxis and epimorphosis. Regeneration is regrowth in the injured region. Regrowth in the injured region. What is regeneration? Regrowth in the injured region. Please remember it. Regeneration was first studied in Hydra by Abraham Tremblay in 1740. So this is 1-mark question. Please underline and take note of the year also. So they may give it along with the year, and they may at one place change the year as 1960, like that. Regeneration are of two types. One is morpholaxis and epimorphosis. You see, the last part of the Hydra produce both the oral end and the aboral end. Okay, so this is called as regeneration. And also you would have seen in the house itself, when the lizard, when another lizard eat upon the lizard's tail, another tail, that is another lizard's tail. Okay, the last part you would have seen, okay, growing as a new tail. Okay, so that is also called as regeneration. Okay. In morpholaxis, the whole body grows from a small fragment. Okay, example Hydra, Planaria. In our 11th book, it is also given. All these examples are also given. Right? So here also I mentioned. Okay, in 12th book, it is given in morpholaxis. The two examples, Hydra and Planaria. Now we are connecting the concepts. Because you are the 12th standard student, you must connect, you must recollect this concept, student. Some members of the platyhelminthes like Planaria show high regeneration capacity. Example, so Planaria belongs to the phylum Platyhelminthes, that is the flatworms, whereas Hydra belongs to the Coelenterata. Example here, Platyhelminthes, under Platyhelminthes, those organisms given as example for this type of regeneration: Tinea solium, tapeworm; Fasciola hepatica, liver fluke; Cystosoma, blood fluke. So all these can be connected with this Hydra and Planaria. So find the group which is correct for regeneration. They can ask. And also I found the diagram from the 11th old book, that is old syllabus book. So this is the diagram to support the regeneration in Planaria. Okay, in our book Hydra is given, very colorful diagram of Hydra is given to understand, and this is also for understanding the Planaria. And here also this is the 11th standard content given. It refers the high regeneration capacity of the that is multicellular triploblastic animal Planaria. Likewise, it is given. When Hydra is accidentally cut, okay, into several pieces, each piece can regenerate the last parts and develop into a whole new individual, student. So we are going to see here, you see, it is cut into 3 parts. Okay, so the last part is regenerated. That is one of the fantastic point that you have to take note here. This is the oral end. The mouth end is called as oral end, student, and the basal end is called as aboral end. So when mouth end, oral end cut off, it knows how to regenerate the aboral end. The same way aboral end regenerate the oral end. If it is middle part, so both sides are regenerated. So that is the point to be noted here. Even sometimes it is cut at the longitudinal fashion. Again, from the longitudinal each piece, you see this side one hydra and this side another hydra is produced. So parts usually retain their original polarity. What is polarity? Just like that you cannot learn what is polarity. You have to ask the question and find the answer. Then only you can remember and you are able to answer very critical question in the MCQ with oral ends. Okay, so this is called as polarity. On one side it is the oral end, other side it is the aboral end. Oral, aboral. So it knows which part is lost. That is called as establishing the polarity. Okay, with oral ends by developing tentacles and aboral ends by producing the basal disc. I already told you to differentiate the basal disc with the pedal disc. Epimorphosis. Epimorphosis is the replacement of lost body parts. Replacement of lost body part. It is of two types, namely reparative and restorative regeneration. Now let us see what is reparative. In reparative regeneration, only, this is the point to be noted, the difference can be asked and you have to differentiate between these two. Only certain damaged tissue, but here severed body parts, cut off body parts is regenerated, but here the damaged tissue produce the same tissue again. That also only certain damaged tissue, not all tissues in an organism. Certain tissue, not all. So this is your understanding. This for your understanding can be regenerated. So here example given for restorative regeneration. Example starfish and tail wall of the lizard. And here you see one arm of the starfish is lost. It is regenerated again. So this is called as restorative regeneration. Okay. So the power of regeneration, this is do you know part. Sponge when macerated, it is cut off into many pieces. That is called as macerated. And squeezed, it means filtered through fine silk cloth. What happens? The cluster of cell which is passing through the silk cloth, okay, they are able to regenerate. That is from the filtrate. From the filtrate, the cluster of cell pass through. It means the filtrate. Regenerate into a new sponge. Okay, this technique is used for cultivation of sponges. Okay, so far what we have learned, we learnt about budding, we learnt about regeneration, fragmentation, gemmule formation. So all these four we learned. Sporulation already learnt in video 1, student. Thank you very much for watching. Wait for the question paper. You will be getting it by tomorrow or day after tomorrow. You are able to answer. All the best for your test. Thank you.